Okay everyone, today we will be doing dissection of bean seed. So here I have a bean seed for you to study. So what we are looking here, let's study the morphology. It's a kidney shaped seed, and let's do these two structures which are visible externally. One is this is hilum. This is the point from where the seed was attached through placenta to the wall of ovary, and this is the micropyle. This was the gap between outer and two integuments through which before the fertilization the pollen tube entered, and now once the seed has been formed, this is the point from where the water enters inside the seed when the seed is soaked in the water. So what we are going to do is we are going to remove the seed coat, which is actually made from outer and inner integument. So let's remove it. This is something different which normally you don't see in the bean seeds. This is actually a radicle which has protruded out. So this particular seed was kept inside the water overnight. So I'm going to make a incision from the micropyle. Make an incision and I'm removing the seed coat. So as you can see, I'm removing this seed coat. Yeah. So the seed coat was made of outer and inner integument. So outer made testa and the inner made tegmen. So testa in this case is looking this brownish structure, whereas in case of tegmen is a very thin filmy structure, which is let me see if I can make it visible to you. Just give me a minute. Let me see. It can be made visible to you. I don't want to damage the radicle while I'm in the process of dissection of the seed. Okay, so as you can see, I have removed the seed coat, the testa with the tegmen. If you want to see the testa and the tegmen, sorry, I will not be able to show the tegmen here. Okay, moving on to the structure of seed now. So here what you are seeing, this are the two cotyledons. These are the two cotyledons. These will be actually the first leaves of the plant when the plant grows. If we put a little pressure, it splits into two, and we see the two cotyledons, one cotyledon and the second cotyledon. In the second cotyledon, the embryo is present. So I'm going to isolate the embryo for you. So before isolating, let me show you some parts of the embryo. This is the part which will form the shoot. If you can see, it looks like the leaf. So this will form the shoot. So this part is called the plumule. This part will form the root, is called the radicle. This is the point of attachment of the embryo with the cotyledon. This, because from here the embryo is attached with the cotyledon. So move it. Yeah. So you can see the embryo, the plumule and the radicle. There are two more parts, epicotyle and hypocotyle, but unfortunately you will not be able to see them here. So we will not be talking about here. We will be talking about them when we will talk about the germination.